Hi all, Dr. Clark here again, and today we're going to talk about genetics. Okay, so this is kind of going to be a overview or a little bit of data collection so you can complete the genetics lab, if that's what you're doing online, if you're doing genetics or whatnot. The first part of the genetics lab, which you'll see starts on page 99, is to just flip coins. Okay, and I'm not going to go through how to flip coins. It's pretty simple. Take two coins, fair coins, flip them at the same time, and you can get the data for that. The second part, or the second procedure, is to examine albinoism. Again, this is just hypothetical. It's just calculating ratios. It's really simple stuff. I'm not going to go through that. The third procedure is to examine hybrid corn. So this is the corn that we typically consume at the store. Well, this one's dried out, but this is what most people are after. Okay, yellow round kernels. Okay, well, that is not the dominant trait for corn. The actual dominant color of corn is purple, okay, or reddish purple. And the recessive trait is yellow. So here you can see this corn right here. It shows you two traits. It's called a monohybrid cross. Okay? And you can see that you have yellow kernels and purple kernels. Well, I've actually went through and calculated, or counted up, not calculated, but counted up the number of purple kernels and the number of yellow kernels. Okay, so there's 211 purple and there's 190 yellow. Now, what you need to do is, in this lab, you need to calculate the probability or the likelihood that you can predict what the parents of that corn cob were. And you do this by making a Punnett square. Okay? You do this by doing a monohybrid cross and a Punnett square. For example, let's say you think that the parents are heterozygotes. Both of them, mom and dad corn, were both heterozygotes. So they had a dominant character and a recessive character for color. If you cross heterozygotes, you have to see, well, what will the ratio of color be? With purple being dominant and yellow being recessive, if you cross two heterozygotes, you would expect that 75% of your kernels should be purple and 25% should be yellow. Okay? So if examining this corn, you think that is the result of two heterozygotes, okay, then that's what you're going to go with. You're going to take 211 plus 190, okay, and then you're going to find out what 75% of that is, okay, and that's how many should be purple, and you're going to find out what 25% of that is, and that's how many should be yellow. You're going to compare those numbers using a chi-square, just like you compared the numbers using the chi-square in the coin example, or I did it for you in the coin example, where if we're flipping a coin and it's a fair coin, we'd expect it to be 5 heads and 5 tails if we're flipping it 10 times. But in actuality, when we really flip it, we get 7 and 3. Are those two numbers different? And we find out that no, they are not statistically different. Okay? It's going to be the same thing for predicting what the parent of this corn is going to be. If you think it's a 75-25, okay, then you're going to use your expected numbers as that 75% of the total, and 25% of the total, and your observed is the actual counts, 211 plus 190. The same thing is going to be true if you think, well, that's not a cross between two heterozygotes. Maybe it's a cross between a heterozygote and a homozygote recessive individual. So you would, if you did that Punnett square, you would expect 50-50. You would expect 50% yellow and 50% purple. Okay? So you're going to do that. You're going to calculate a chi-square. You're going to examine the chi-square table in the back of your lab manual, and you're going to see if statistically you are correct in your guess of the parents of that ear of corn. If the numbers are statistically different from each other, which means the observed and the expected would be different from each other, then you are wrong in your estimate of the parent or the parental genotype of that piece of corn. If there is no difference between the observed and the expected, then you are correct. Okay? And that's a pretty simple method to do that. All right, the next one is a little bit more difficult because this is a dihybrid cross. And dihybrid cross means that we're dealing with two different traits. Okay? In this case, we're dealing with color. And again, purple is dominant and yellow is recessive. Okay? But we're also dealing with shape. Okay? So either the kernel is plump and round, or the kernel is wrinkled. And so hopefully you can be able to see that on the video. You can see that there's wrinkled kernels and round kernels, and both yellow wrinkled and yellow round, purple wrinkled and purple round. Okay, so I already went through and gave you the numbers. So purple smooth or purple round, there was 196. Purple wrinkled, there was 97. Yellow smooth, there's 125. And yellow wrinkled, there's 61. Okay? Again, just like what you did before to calculate how many or what your expected values should be, is you have to add those numbers together. 196 plus 97 plus 125 plus 61. That's your total kernels or total offspring. You take that, okay, and then you can divide each number by the total. So 196 divided by the total, that'll give you the percent purple smooth that you found, okay? And then the percent purple wrinkled, etc. If you want to find out how many you would expect, then you need to find out what ratio for the dihybrid are you going to deal with. Do you think it was two heterozygotes crossed? If you cross two heterozygotes that are dihybrid, then you get a 9 to 3 to 3 to 1 ratio. So you would have 9 purple smooth to 3 purple wrinkled to 3 yellow smooth to 1 yellow wrinkled. If you take 9 plus 3 plus 3 plus 1, that equals 16. Okay? And if you take 9 divided by 16, it's going to give you 56%. So you would expect that 56% of the kernels, however many there were once you've added it all up, that's how many you would expect to be purple smooth. Okay? And so you're going to go through that and do it for purple smooth, purple wrinkled, yellow smooth, yellow wrinkled, and you'll get your expected values and your observed values. And then again, you can do a chi-square analysis and see if you're correct. If there's no difference between the observed and the expected, then again, your hypothesis or your guess on the parental genotype is correct. If there is a statistical difference, then that means you're wrong and the parents were something other than what you said. All right, the last piece is going to be class data. Now, if you're doing this assignment at home, clearly you're not going to know the class's data on whether or not they're going to be dominant or recessive, okay, for certain characteristics, human characteristics. And so what you need to do is you need to find 10 people and find out, do they have attached earlobes? Do they have bent little finger? Do they have a hitchhiker's thumb? Interlacing fingers, mid-digit hair, pigmented irises, and widow's peak. Now, please, if you're doing this at home, do not use 10 family members. Okay, so if you can find 10 family members, don't use 10 family members, because certain traits might be more likely to pop up in your family. Maybe, for example, your mom and your dad had blue eyes, okay? And you have blue eyes, and your brother has blue eyes, and your sister has blue eyes, and your other brother has blue eyes, and on and on and on. Okay, well, that's not looking at a diversity of people. Okay, you're going to see that a trait, when you're examining a single family, a trait can look like it's kind of fixed in that family. Okay? And we don't want to do that. We want to look, say, if you're picking randomly, maybe you pick a couple neighbors, you pick a couple people at work, a couple family members. Are any of these traits more likely to pop up as dominant or recessive? Okay? And is being dominant, more likely to occur on traits in a population? And that's what you're trying to answer. Okay? And so you can go about by examining people's ears and their fingers and things like that to see if these traits are dominant or recessive. Okay? If you're just reviewing this because you're going to come to campus and do this lab on campus, then we'll collect data on the class when you come to campus. Okay, so that's pretty much it. Again, your chi-squared table is in the very back of the book. All right, so when you calculate your chi-square value, you need to examine the table that's in the back of the book. To examine the table in the back of the book, I'm going to show you real quick how to read the table. Okay, so here's a chi-squared table. And let's say we're analyzing this corn. So this corn, there are two possibilities. Right? So your trait is either purple or yellow. Right? So you're going to take that 2, and to get your degrees of freedom, or your DF, you're going to take n minus 2, or n minus 1, and n is 2, because you only have 2 possibilities. So your degree of freedom for that monohybrid is going to be 1. So you're going to go over to 1 on the table. And in biology, we always use 95% confidence. So we accept that there could be 5% error on our experiment. Okay, so we're going to take, and we're going to look 1, and we move over, and the value on the chi-square table is 3.84. If your value that you calculate for the chi-square is bigger than 3.84, then the two things that you're comparing are statistically different from each other. If it's less than 3.84 in this case, then the two things that you're comparing are statistically identical. Okay? So we're not saying that there isn't a difference between the numbers, but we're saying statistically speaking, there is no detectable difference between those numbers. Okay? In the case of the dihybrid cross, you have 4 combinations. You're going to go to 3, still under 0.05. That number would be 7.81. Okay, your value is either going to be bigger than that or smaller than that, and that's how you read the chi-squared table. Okay. All right. So I hope that helps. If you're going to do this at home, and you need help, clearly, obviously, email me or call me. If you're coming to campus to do it, I'll see you on campus and I'll answer any questions at that time. Okay. All right, until next time. Last time, I verbally told you how to do the chi-square, and now I'm going to visualize it or show you a visualization of using the chi-square to predict whether or not you can accurately tell the phenotype of the parents of a piece of corn, or it doesn't really matter what we're talking about. It could be human parents, it could be really anything. Okay? If you know the phenotype of the children, and you know that that phenotype is controlled by a single gene, then you have the capability of figuring out the genotype and phenotype of the parents. Sometimes not the genotype, but definitely the phenotype. Okay? All right, so let's go through this. Let's take this one first, okay? Let's say that we think the parents are 2 heterozygotes. Okay? So we do the Punnett square,
Okay? And we get a genotype or a genotypic ratio of 1 homozygote dominant, 2 heterozygotes, 1 homozygote recessive. The phenotypic ratio, because there's no incomplete dominance or anything like that going on, the phenotypic ratio means that you have 3 dominant for every 1 recessive. Okay, a 3-to-1 ratio. Knowing that, we can always say, well, what's 3 out of 4? That's 75%. So we would expect that 75% of all the offspring that are produced by a cross of 2 heterozygotes, 75% of them are going to be dominant, 25% of them are going to be recessive. Whatever that trait might be. Maybe it's eye color, maybe it's kernel color, maybe it's hair color. There's all kinds of traits out there that could be based on these dominant recessive genes. All right, so let's just take corn for example. Let's say that 75% is going to be purple and 25% is going to be yellow, and that's our prediction. We have 210 offspring. Okay? So therefore, out of 210, we would expect 157 to be purple and 53 to be yellow. Let's say we went out and we actually counted a ear of corn, okay? And the numbers that we get are 138 and 72. Okay? So we can do a quick chi-square analysis to see, is our prediction that the parents are both heterozygotes correct? Okay? And we do that by using this equation. Okay? This is not x squared, this is chi squared, okay, is equal to the observed minus the expected squared divided by the expected. Very simple equation, very simple statistical analysis. Okay, so we've done that. 138 is the observed purple kernels. 157 would be the expected purple kernels. Square that, divide it by 157. You're going to add that to your other option. Your other option is yellow. So we found that there was 72 yellow kernels. We expected that there's 53. Square that, divide it by 53. And this is our chi-squared calculation. Once you add the two together, the answer is 9.1. So then if we go to the table, and remember that we're looking at 95% confidence interval, or an alpha level of 0.05. So on the table, you want to look at 0.05. We have 1 degree of freedom because we only have 2 choices, purple or yellow. If we pull that number, it's 3.8. Okay? If your calculated is bigger than the table value, then the two things that you're comparing, the observed and the expected, are statistically different from each other. So in this case, that means that we are incorrect. Okay? The parents of that cross are not heterozygotes. Okay? We can do this the same way, but use a different parent cross. So maybe we think one's a heterozygote and one's homozygote recessive. We do the Punnett square, we figure it out. The genotype is a 1-to-1. There are, 2 heterozygotes for every 2 homozygote recessives, so it's a 1-to-1 ratio. You get 1-to-1. The phenotypic ratio is the same, dominant to recessive, so we're expecting a 50-50. So your expected, if you have 210 offspring, your expected is 105 for each. Our observed is the same, because we only have to count the ear one time. We got 138 to 72. We do the math, the observed minus the expected squared divided by the expected. Our calculated is 20.8. Our table is the same. And again, we are incorrect. It is not a cross between a heterozygote and a homozygote recessive individual. This is the way that we can test predictions. Okay? And we talked about this before that statistics allow you to analyze data in a very meaningful way. We're analyzing hypotheses about what the parental type is, or what the genotype of the parents of offspring that we know what their phenotype is. Okay? Now, what might have happened, and this happens all the time, when we do this, we find out that we were wrong. The color of a kernel is not a single trait, or the color of a human's eye is not based on a single gene, but rather it's multiple genes. And one way we can do that is by setting up a Punnett square, making predictions, counting offspring, and finding out statistically we're wrong. Statistically, this is not possible. That means that maybe it's not a single gene, but it's multiple genes. Okay? So that just gives you kind of an overview of how to do that. You can use the data that, please use the data that I gave you, from my corn counts, because your values and your statistics are going to be very different than what I did on the board. What I did on the board was hypothetical. I just made it up. Okay? So the other ones, you can just use the data that I actually collected. Okay? All right. Have fun.